Hi, so welcome to another video in the topic-wise question of biology series. So this topic is about respiration muscles and internal environment. Now let's begin. The first question asks, the diagram shows the sequence of electron carriers in the transport chain. The diagram also shows the stages where ATP is generated. So reduced NAD first gives a proton to the NADH dehydrogenase, which then gives the proton to coenzyme Q. The proton also comes from reduced FAD to the coenzyme Q, and also in this part from where NADH dehydrogenase gives a proton to coenzyme Q and ATP is formed. Then we have the cytochrome B, which accepts hydrogen from coenzyme Q, and this donates the proton to cytochrome C1. This produces 1 ATP, and then cytochrome C1 gives protons to cytochrome C, and so on. The electron transport chain carries on further, and our last electron acceptor is oxygen. Okay, oxygen, and this produces a final ATP. So in this total cycle, we get 3 packets of ATP. So let's read the first question. Where is the site of electron transport chain? So do we remember where is the site of electron transport chain? It is in the inner mitochondrial membrane, right? Then let's move on to the next question. The table gives some information about inhibitors of the electron transport chain. So we are giving inhibitor cyanide and rotenone, and these are the sites of inhibition. Our first question asks us, explain the effect that cyanide will have on ATP production by the electron transport chain. This is for 2 marks, and we have to explain the effect that cyanide will have on ATP production. So first of all, if cyanide prevents from donating hydrogen atoms to the individual complexes, and so the first thing that we can write here is no ATP will be formed. No ATP will be produced. Will be produced by the electron transport chain. You will get 1 mark for answering this one. And our second point is will be this is because electrons passed onto oxygen. So we remember that cyanide affects the last complexes, which gives electron to the oxygen atom, which accepts the electron. As a result, the electron transport chain is completed. But since the complex before the oxygen is now inhibited, the rest of the electron transport chain is also inhibited, and the electron transport chain will not be completed. As a result, the electrons will not be passed on to oxygen, and so there will be no ATP produced. Are we clear? And lastly, we can write therefore the electron transport chain will stop. Alright, so I wanted to ask you that to specifically remember the electron transport chain and how all of this occurs. This is the topic on ATP production. Keep this memorized. You have to memorize it very well, as this type of questions often come in the exams. And if you are not familiar with this type of question, then you might want to revisit the topic of ATP production. You will find that playlist in the channel in my description box below. Okay, so the next question is metabolism of succinate results in the production of reduced FAD. Explain the effect that rotenone will have on ATP production by the electron transport chain. So firstly, we have to write the effect of rotenone on ATP production. The first point would be there will be no ATP production from reduced FAD. Reduced FAD. Okay. Sorry for the mistake. It would be reduced NAD. Reduced NAD. Okay. But I'm coming to the explanation in a bit. Let me write this first. But there will be some ATP reduced from reduced FAD. Because electrons from reduced FAD enter. So quite a long answer. And let's write ETC, short for electron transport chain. ETC after the site of inhibition of rotenone. Okay. Now let's read the answer again. There will be no ATP production from reduced NAD. Because, okay, let's get to the chain again. Okay, What does rotainen do? Rotainen inhibits the site between NADH dehydrogenase and coenzyme Q. So basically what rotainen does is he inhibits this site, okay, from NADH dehydrogenase to coenzyme Q. So here we have seen that this exchange produces an ATP, but since now this is inhibited, this ATP will not be formed. So, and this also forms because the reduced NAD gives the protons to NADH dehydrogenase, which transfers to coenzyme, so hence ATP is made. But since now this part is inhibited and NADH dehydrogenase does not accept any protons from reduced NAD, hence this ATP will not be made. So there will be no ATP production from reduced NAD. Okay, but there will be some ATP produced from reduced FAD. Okay, because this part right over here is inhibited, right? But reduced FAD still gives a proton to coenzyme Q, right? This coenzyme Q still gets a proton from reduced FAD. So then this chain continues. This chain continues to go, and then our final oxygen accepts an electron, and thus at least some ATP will be made. These two ATP will be made. So that's why we have said in our answer that there will be some ATP produced from reduced FAD because electrons from reduced FAD enter the ETC after the site of inhibition of rotainen. Alright? Okay. Now the next question asks, explain why these inhibitors will not affect the production of ATP from anaerobic respiration. Use the information in the table to support you. Okay. So this question asks us that these inhibitors will not affect the production of ATP in an anaerobic respiration. We have to explain why, and we can use the information in the table to support our answer. This is for 2 marks. So first of all, we will write that the inhibitors actually affect the electron transport chain. Inhibitors affect electron transport chain. Which is not used in anaerobic respiration. Used in anaerobic respiration. There is no ETC in anaerobic respiration. This is first point. So the inhibitors have no place to inhibit the electron flow of electrons. Okay, so that's why it doesn't affect anaerobic respiration. You get 1 mark from this. Then, therefore, the same quantity of ATP will be produced from substrate level phosphorylation in glycolysis. Okay. So what this essentially means is that the same number of ATP that is formed in anaerobic respiration, it will remain the same from substrate level phosphorylation of glycolysis. Phosphorylation in glycolysis. Are we following up till here? Also, pardon my handwriting. It's not very great, but sorry that you have to bear with it. Okay. Let's move on to the next question. Okay. Question number 2 says, the kidney is involved in osmoregulation and the production of urine. Which substances are filtered from the blood in the renal capsule? So we have to choose the correct answer. Which substances are filtered from the blood into the renal capsule? So remember, that is going to be glucose and urea. Okay, and then 2 asks us which structure is acting as the filter in the renal capsule. That is obviously the basement membrane, which acts as the filter in the renal capsule. And our 3 asks, which transport mechanism is responsible for the uptake of glucose into the cells of the wall of the proximal tubule? So we have to choose the correct mechanism from these options, which is responsible for the uptake of glucose into the cell of the wall of the proximal tubule. Okay, so this is sodium cotransport. The mechanism is sodium cotransport. Now we come to the next question. Explain why some urea is reabsorbed in the proximal tubule. And here we have to explain why some urea is reabsorbed. This is because the concentration of urea is greater in the filtrate than in the blood. So since there is a greater concentration of urea in the filtrate than in the blood, urea will diffuse down its concentration gradient. Remember what is diffusion? So when there is a lower concentration gradient, the particles move from the higher concentration gradient to a lower concentration gradient. Okay, here urea will diffuse down its concentration gradient Okay, you can totally write it up to here. And also remember, they will be easily diffused down the concentration gradient because the walls of the proximal tubules and capillaries are permeable to urea. Okay, clear? So let's move on to the next question. In an investigation, salt solution was injected into the carotid artery of a mammal. The carotid artery carries blood to the head. The rate of urine production was measured after the injection. The table shows the results. So salt solution was injected in an artery of an animal. The artery carried blood to the head. The rate of urine production was measured after this injection. Okay? And here is the table where you can see. We have to calculate the percentage decrease in the rate of urine production 10 minutes after the injection. So we have to calculate the percentage decrease here of 10 minutes after the injection. Okay, so this one. So firstly, we have to subtract the values of the first one. So at time 0, our rate of urine production was, we have to find out the difference from time 0 to time 10. Okay, difference would be 7.0 minus 0.6, and this gives us a value of 6.4. Now that we have the difference, we can easily find out the percentage decrease as well. Just minus 6.4. You divide it by 7 and into it by 100, and then you will get 91.43, which is the percentage decrease. Right, we can totally give any here. It's advisable to keep it in a significant figure. Let's move on to the next question. At what times should more measurements be taken to find the lowest rate of urine production? We have to see the data above, chart above, and we have to answer that at what times should more measurements be taken to find the lowest rate of urine production. So our answer would be B, at 1 minute intervals between 5 and 15 minutes. Between 5 and 15 minutes. 5 and 15 minutes, because if you see, the urine production between 5 and 15 minutes is the lowest. Okay. Now we have to explain the results of this investigation using the information in the table. Okay. Now we have to explain the results of this investigation. So what happens is, basically the investigation is about the urine production per minute after injecting the salt solution in the carotid artery. So we have to explain it after what happens, the injection. Okay, after what happens after injecting the salt solution. So basically we know, we have read the carotid artery carry the blood straight to the brain, right? But we have to be specific here, which part of the brain. So we have to write where does it take the blood? To the hypothalamus, straight to the hypothalamus of the blood. of the brain. Hypothalamus of the brain. Of the brain. Alright? Hypothalamus of the brain. Now, since there is a change in the blood and it's straight up in the hypothalamus of the blood, what will happen is hypothalamus will detect. It will hypothalamus, by the way. It will detect the lower solute potential of the blood. We know that since the salt solution is now very concentrated in the blood, there will be lower solute potential. So this will be detected by the hypothalamus since blood is directly traveling from the carotid artery to the hypothalamus. As a result, the pituitary gland will release ADH. And then ADH will result in the absorption of water, so less urine is produced. Okay, ADH will in more reabsorption of water, so less urine is produced. And then this is the main 4 points that we have written, which you will get marks for. But you can also know that what happens next is, so then now since water level is balanced again and less urine is produced, so the solute potential returns back to normal and it is balanced. ADH will no longer be released because it is the solute potential now is balanced. And then urine production will go back to normal again and it will increase. So that's it for this question, and this is the end of the video. I hope it's clear what I have said till now. If you have any problems, feel free to reach me out or comment down below. Thank you for watching the video, and I will be back next time.